Hi, my name is Yanni Pandis, and I'm going to be presenting the molecular biology avatar. I'm working at Professor Grove's lab at Imperial in the context of Etrix. And okay, so when we were discussing the structure of this agenda with Kays, we said that someone has to present the molecular biologist avatar. As I was told by my supervisor, I'm not the molecular biologist. Okay, fair enough. I accept that. 5 years of PhD won't do that. And of course, if you haven't had any experience, you can't call yourself a molecular biologist. But okay, so what I wanted to really to say is just show you what the work I did, well, some of the work that was done in the PhD, and it's running its own. I just wanted to put down all of the work that okay, yeah, as I said, I was doing at the PhD, and how when I finished this project and talking to Anthony Rowe, this is the reason why I came into the whole Transmart and Etrix development team, because this is it was like, how can we make a system do all this work together? So just
Not working?
which is the pointer?
There's an arrow
Here you
to.
go.
Oh, sorry.
Yeah, I think I'm controlling it now,
Yeah.
right? Okay. So the motivation between this research was initially, I was working in a rheumatoid arthritis animal model laboratory, and we started off trying to assess the microRNA involvement in rheumatoid arthritis. But at the same time, we were as part of an IMI project. We were collaborating with clinicians, and we had samples from both mouse and humans, and we just tried to see how we could do this in a unified way. So the idea was to try and find novel biomarkers and drug targets, as pretty much I think the target of any IMI project would be in a specific disease area. So what are microRNAs? I'm not going to go into this at all. Essentially, small regulatory RNAs that they bind to a target mRNA or a gene after it's been transcribed, and they lead to its blockade of being expressed. So if it binds, for example, to a gene called TNF, this microRNA will inhibit TNF's expression. So you'll be able to see in most of the cases in the first very few seconds of its being born, you'll be able to pick up a transcript. So if you put it on a microarray platform, you'd see it lighting up. But minutes after that, when it's been bound by this microRNA, it's starting to get degraded, and you can either see that at the mRNA level or at the protein level. So the first thing that we had to do is we had to set up the experimental design. So you need to talk about what sample sources you're going to be using. You're going to be using a rheumatoid arthritis patient. You're going to have osteoarthritis patients who are healthy controls to be able to compare against. And the same in this case, we use. So what we wanted to do is we wanted to extract some plasma. The reason we chose plasma is that it's been shown that microRNAs were very, very stable in plasma, and they can be indicative, and they can be used as a biomarker. So this is the whole biomarker potential here. And then we just chose to take a couple of cell types which are very specific and known to be detrimental in the disease. So synovial macrophages and fibroblasts, which have been the focus of research many years. So what we wanted to do is do some high throughput profiling, specifically some microRNA sequencing, to try and determine the profile in different cases for disease versus normal. Do a bit of microarray-based gene expression profiling, proteomics maybe, and a couple of bioinformatics and analytical methods in here. So so what I would like to do, if I had a system next to me that could record all this information, could help me along the way and not having to use a plethora of different programs to do this, you want to get all the information, the clinical and the phenotypic data, such as, for example, disease onset, severity, histopathological data, into a database. So somewhere which is recorded and not having to receive the Excel spreadsheet from someone. Do the same for the animal models. Okay, clinical data might not seem relevant to an animal, but you can tell how severe its disease is by simply looking at its paw and seeing how much it's swollen up. You can also get him histopathology data from that, and well, disease onset probably is not pertinent. Um, for for the plasma data, you need to know what extraction protocol you use specifically to extract the plasma. So I want this to be recorded just so I can go back to it. I like to know the extraction volumes. I like to know, and then when I'm producing the my samples, I need to know how good my RNA was that I'm going to profile. So I'd like to know the RIN number, and I'd like to have some sort of some sort of measure on the proteins as well, just so I know that I'm analyzing something which is good, so I'm not going to get crap data out of it. For the cell data, it's very important to know again what the isolation or extraction protocol, how I isolated these cells, how I cultured these cells, because this you can come back to this and it can bite you if you don't have the right information. The cells will become senescent, and you also again need some data on the integrity and quality of the samples you have. So I want to collect all of this information and finally put it into a database so I can get it back whenever I need to. So then what the next thing I'd like to do is I'd like to generate the microRNA profile. So I'm going to take not only microRNA but also the others as well. So I'm going to take my samples, extract the RNA, and I'm going to put them on the sequencer. And first thing that I'll get is, and I understand it's just a long string of sequence. It means nothing to me. The bioinformaticians are going to come in and do their own tricks. Hopefully, I could do that by a couple of buttons if it's standardized, and I want a quick method to do this. I'd like to pre-process a line and normalize the reads that I'm getting from for each microRNA. I'd like to do the same thing for the microarray, the gene expression. I mean, it's been so many years that we've been working on these things, and as Kay said, there's so many platforms to do microarray expression analysis. It should be done automatically now. We shouldn't be this shouldn't be the focus. So I need to press a button and it will pre-process and normalize my samples. I like to do the same thing for my proteomics, and then I want to generate a profile, specific profile of disease versus control versus normal. I'd like a gene profile for mice and men. I'd like a microRNA profile, and I'd like a protein profile. I'd like to store all this on a database. So whenever I come in, I can ask it straight away to tell me what I'm looking at. So then one of the most interesting things would be trying to see which animal model would best describe the patients I'm looking at. So once I've decided which cohort or which subset of patients I'd like to further analyze, I'd like to take the microRNA profile from this patient or from this patient group, and I'd like to get the same thing out of my mouse. So then by comparing, I'd get these Venn diagrams and I'd find the intersection, which would mean that this is something which is conserved. So what I'm seeing in humans, I'm seeing in mice as well. Perfect. This is what I want to work on, and this is what I'm going to conserve, called the conserved. So conserved between the two, or throughout evolution between the two species. I'm obviously going to validate this independently in a separate method, and I'd like to record all this information in the computer as well. But you know what? I checked a list of 100 conserved microRNAs here. Out of these, I could validate the 50, and this is what I'm gonna end up with using in my and storing. So, as I said, you have this. Okay, so then you go on to, for microRNA specifically, you need to go on to try and predict what targets, what your microRNA is influencing, which genes. As I said initially, it binds to a gene, but it can bind to multiple genes and influence its regulation. So what I'm going to do there is I'm going to go out through a click of my button, and I'd like to check what microRNAs, the microRNAs in my profile, what genes are they targeting. So I'm going to get a list, a big list of genes which are target for my own microRNA. Again, I'm going to do the same thing. I want to find the intersection. So I'm going to look at the conserved predictive targets of my disease and store these in information. So what I'd like to do then is, through systematically, I'd like to compare the targets that I found against my gene expression and my protein expression profiles, just to see what really holds true. So I've predicted that this microRNA may bind and may influence this gene. Can I have a look and see on my computed on my calculated data if this exists, if this interaction? Again, I'm going to end up with something which is validated in a conserved microRNA target and put it into the database. One of the things when we were discussing this back in the day with Anthony was maybe to think about converting the mouse genes and proteins to their human orthologs, just so you base them. But again, this is something done individually. You might want to do this systematically next. Finally, the final thing I'd like to do is, guess what, a functional enrichment analysis, whether that's going to be on gene sets, whether that's going to be on ontology terms, whether that's going to be on a pathway. What I want to do finally is do this comparison and store it on the database so I can see exactly what I might be influencing. And that's the end of my presentation.
Thank you, Yanis.
Any questions?
I ask you a question.
Mm-hmm.
You're generating a lot of data, aren't you? So if you have data retention policies for each step, and what you keep and what you don't keep and where you keep it.
So in the scenario I just described, I'd like to keep the
typical reaction from molecular biology.
well, I think Ian was describing. I think he's probably going to ask me, do you want to store the raw reads from the sequencer versus the process files? Okay, I can live with and with storing the outputs of all these procedures, but I would like the bioinformaticians I'm working with to be able to access it at some point.
It's not immediate access.
No.
I think I have a similar question, and that is, which part of your story do you see should be accommodated by transport?
So in this story, I just try to describe a general scenario, and then thank you for the question. Then so ideally, I'd like the entire thing to be handled by Transmart. Ideally.
Realistically.
Realistically. Okay. Realistically. This information definitely. So the records. Okay, I'm biased at the moment. It's really hard to ask me because I know what it can do. Okay. So this information I definitely want to make sure that it's the profiles. I definitely want them to be stored. The now all of this kind of I would like a calculator which would tell me what's the same, but maybe okay, this part leave it out. If I could input it back into the system once I've calculated, that would be fine. This definitely not in Transmart. This would be I need a button or an export for my list to go out and do this to whatever database I want to use. So this is the microRNA target prediction. Why reinvent it? And I'd probably store the conserved targets again, as long as I can have access to these, so I know I can send my collaborator to find them. I'd like to store what I've validated, and probably the outcome of this result. I don't think it is everything.